This is chapter 21, the immune system, part 2. As a review, what we talked about in part 1, we started with the innate defenses. The first line of defense are the surface barriers, the skin and mucous membranes you see here at the top. Then you have the internal defenses, your phagocytic cells, your natural killer cells, inflammation, antimicrobial proteins, and fever. And then we were starting with the adaptive defenses, talking about B lymphocytes that are important for humoral immunity, and then your T lymphocytes, which are involved with cellular immunity. And we were talking a little bit about this third line of the defensive system of the body, which is very specific, it's systemic, and it has memory, which the first two are innate and they're non-specific. So T lymphocytes are important for a cell-mediated immune response because antibodies are useless against intracellular antigens. T lymphocytes are involved with mounting up a defense against intracellular pathogens. Now, there are two populations of T cells based on which glycoprotein surface receptors are displayed. So you see here you have what are called CD4 cells. They're also called T helper cells. And these T helper cells are very important because they activate B lymphocytes, other T cells, your macrophages, and they direct the adaptive immune response. And you're going to hear me refer to these cells as the general, because they are in charge of getting the whole immune system mobilized to attack a particular pathogen. Now your CD8 cells, which are another type of T lymphocyte, these become cytotoxic T cells. And so CD8s are the same as cytotoxic Ts. These can actually destroy cells harboring foreign antigens, and they also become memory T cells. So antigen recognition. This provides the key for the immune system to recognize the presence of intracellular microorganisms. So there is a complex that's on all your body cells, and this is called the MHC complex, or major histocompatibility complex. Now, if your cell proteins or these cell markers they normally are ignored by T cells if those markers are complex with self-protein fragments or normal proteins made in the body. So your T lymphocytes are not going to attack your cells because those cells have markers that define those cells as belonging to self. T lymphocytes are programmed, remember, to attack anything that's considered non-self. So your MHC proteins are ignored. And immunocompetent, those are the ones that have gone through the training. Immunocompetent T cells are activated when they bind to a recognized antigen that's displayed by an antigen-presented cell or an infected body cell. So you have these cells that are called APC cells. They ingest something that's foreign, and then they put it up on their marker, and then they present it to B lymphocytes or T lymphocytes to let them know that there's a problem going on and that they should mount up a response against it. Then you also have a situation where you have a body cell that's infected, something going on inside. And these T lymphocytes are able to recognize a foreign protein displayed on that antigen of that particular cell to alert it to destroy that cell that is infected. So T cells must be able to simultaneously recognize the non-self antigen that is in the complex of the MHC protein. So they have to recognize the non-self part, and they have to recognize the self part. And the self is the MHC protein of a body cell. And the non-self is usually displayed within that MHC complex on your cells that belong to you. So these MHC proteins, these major histocompatibility complexes, There's two types that are really important to T cell activation. The first one is class 1, and then you have class 2. But let's talk about class 1. Class 1 proteins are displayed on all of your body cells with the exception of red blood cells. That's why red blood cells can be transfused from one person to the next without fear of rejection, because they don't have these MHC complexes. So all your body cells with the exception of red blood cells display class 1 markers. Those are your self markers. Class 2 are displayed by your immune cells. So your antigen presenting cells, your dendritic cells that are found in epithelium and also in connective tissue, your wandering macrophages, your B lymphocytes, they all have these, and that's how they work with each other. Okay, they can coordinate with each other by binding to these class 2 markers. These, remember, on all body cells. Now, the cells that recognize the class 1 and recognize that there's a problem within your body cells are the cytotoxic T cells that we talked about, or the CD8 cells. They're able to recognize if those cells of your body are infected by something that they can recognize that's foreign. Now, your class 2 are recognized by the T helper cells. Now, remember, we said the T helpers are the general. They're the ones that orchestrate, get together the whole immune response. So these are the cells that can recognize and stimulate all these other cells, and it's through these class 2 markers. That's how they work together. So this picture kind of pulls everything out and explains the two different arms of the immune system with the T lymphocytes. According to whether they have the class 1 marker, how that works, and then the class 2 markers. So remember, class 1 is displayed on all nucleated cells. So there's your class 1 marker right there. Now, these class 1 markers are recognized by your cytotoxic T cells. There's your cytotoxic T cell, there's the T cell receptor, there's the CD8 protein. And when foreign antigens on the major histocompatibility complex, it's on your body cells, are complexed with endogenous, these are intracellular pathogens or proteins made by cancerous cells. So you have something going wrong inside, that's endogenous. And then if those proteins become displayed, foreign antigens on the major histocompatibility complex send this message. If displayed by an antigen presenting cell that has ingested that particular pathogen, that antigen presenting cell will say, I belong to self, but I've captured a foreign invader. This is what it looks like. Kill any cell that displays it. If displayed by any other body cell, then the message is, I belong to self, but I've been invaded or become cancerous, kill me. So the idea is to stimulate the cytotoxic T cell to destroy any cells that are harboring this infectious agent, and they have the class 1 marker. If it's class 2, remember, this is how your immune cells are able to crosstalk with each other. So antigen-presenting cells, your dendritic cells, your phagocytes, your B lymphocytes, they ingest something and they display it on their class 2 marker. Then your T helper cells, remember the T helper is the general. They bind to these antigen-presenting cells and co-stimulate them and make the immune system respond in force. Now, these are exogenous proteins. These are phagocytized extracellular pathogens that are digested by your antigen-presenting cells. They're like the Pac-Man. They go around eating stuff and then they present it to the T helper cell. So the message is, I belong to self, but I've captured a foreign invader. This is what it looks like. Help me mount up a defense against it. So remember, this is how your immune cells work together, is through these class 2 markers, and your key player here is the general, and that's your T helper cell that is able to stimulate all arms of the defense against whatever this is that they have encountered, ingested, and displayed on their MHC class 2 marker. This one here, this is all body cells, and they display the class 1. That's recognized by the cytotoxic T cells. So if they encounter a cell that's displaying something foreign, they can destroy that cell. So T cell activation, in order to have the full response of that particular T lymphocyte that has been activated, the T cell has to bind, okay, and it also has to release these chemical messengers. This is called co-stimulation. And these chemical messengers are called cytokines, and they're in families. It could be interleukins 1 and 2, and there's a whole family of interleukins, and I'm going to minimize discussion, but these are just chemical messages that cause the T lymphocyte to produce a clone and proliferate and make an army of itself so it can fight off the infection. So this process of co-stimulation by the release of these chemicals triggers proliferation and differentiation of the activated T cell. So the key is here, if you don't have this, you can have binding, but if you don't have the release of these co-stimulatory chemicals, this process results in anergy. Anergy means nothing happens. And the T cells become tolerant to the antigen, they don't respond, they don't divide, they don't secrete any cytokines, so you get no response. So stimulation is very important. So this picture is talking about clonal selection, just kind of reviewing what we've talked about already. And this is looking at an MHC class 2 marker being displayed on an antigen presenting cell. In this case, it's a dendritic cell. So the dendritic cell engulfs an exogenous antigen, something from the outside that it's come in contact with, processes it, digests the fragments, and then puts the fragment pieces on its MHC class 2 marker protein. Now, the T helper T cell recognizes the antigen, the MHC complex, and they co-stimulate each other. And so the T helper cell binding to this dendritic cell, co-stimulation, and now you get the formation of a clone. So you get a clone of T helper cells that actively stimulate other cells that are immune cells into the attack against this foreign pathogen, and then some of them become memory cells, because remember, with this adaptive arm of the immune system, you do have memory. So some of the cells become memory cells, so if you encounter that pathogen again, you can eradicate it. So the T helper cells, their function cannot be overstated, because they draw in the other arms of the immune system, so they can attract your neutrophils, they can attract natural killer cells, they can attract macrophages, they can stimulate B and T cells to undergo mitosis and mature and join in this fight against the foreign pathogens. Without the T helper, there is no immune response. Very important, because if you knock out the general, the army will not respond. So without T helpers' ability to join into this fight and recruit everybody else in, you're not going to have an adequate immune response. Okay, so cytotoxic T cells. These are the only cells that actually directly attack and kill other cells that are infected. Activated T cells, these cytotoxic Ts, they circulate in the blood and lymph and lymphoid organs in search of body cells displaying antigens that they recognize. Their targets are cells that are infected by viruses, so the agent, the pathogen, is hidden inside. Cells with intracellular bacteria or parasites, cancer cells, or foreign cells that are associated with transfusions or transplanted tissue. This slide's showing what a cytotoxic T actually does, how it attacks infected or cancerous cells. You can see this picture off here to the side. It's showing you a cancer cell, and you have a cytotoxic T that has attached itself to it, and this cancer cell is undergoing apoptosis, which is cell suicide. So what happens is the cytotoxic T cell identifies foreign antigens on the MHC1 proteins, and then binds to that cell. So this could be a tissue cell that has a virus or a cancer or something associated with it, intracellular pathogen. The cytotoxic T cell releases perforins from the granules that are in it. And those perforins are proteins that will assemble and form a hole in the pathogen's membrane or a cell wall. And then there are these proteins that are called granzymes that go into that opening that's caused by the assembly of the perforins. And the granzymes will actually enter the target cell by pores, and then once they get inside, the granzymes activate enzymes that trigger apoptosis. So the assembly of the pore, the release of the granzymes to initiate apoptosis is what causes the destruction of that particular foreign cell or that particular infected cell. So that's kind of how it works. Okay, so we talked about the T lymphocytes. I just kind of want to back up a little bit to humoral immunity response, and that term humoral refers to the humors of the body, and the humors are the fluids of the body. Primarily, when we think about immune cells going through body fluids, usually we think of blood. So this is the job of the B lymphocytes that are going to be involved in producing antibodies. Now, the indirect method of defense involves B lymphocytes producing antibodies that bind to antigens and tag them for destruction. Recognition begins when an antigen binds to receptors on B cells and then is phagocytized, and it requires cross-linking of receptors. Then the ingested antigen is displayed on the cell surface to activate the T helper cells. So you can have this indirect method that involves B lymphs producing antibodies just directly. And then you can have this recognition where the B cell ingests something and presents it to the T helper cells. This is normally how it works. That's the most common pathway, is phagocytosis and displaying it to activate the T helper cells. Then when the T helper cells are activated, they secrete interleukins. These are the chemical messengers that result in co-stimulation. Okay, and co-stimulation is going to activate the B cell. That's going to cause the cloning of the B cell, just like we saw when the cytotoxic T cell was co-stimulated with the helper T cell slide or two back. Okay, so you always have to have that co-stimulation to form the clone. So here it is in picture form, the primary response, initial encounter with the antigen, and you can see the antigen is binding to these receptors on the B lymphocytes, and then they form a clone, and then most of them become plasma cells. So the B lymphocytes will differentiate into plasma cells, and see how they look different? All this inside, this pink stuff, this is endoplasmic reticulum. And remember that endoplasmic reticulum is a manufacturing plant of the cell. And so when they become plasma cells, now they can start cranking out antibodies. And a fully activated plasma cell can crank out 2,000 antibodies per second. They churn them out, and these are what are going to attach to foreign pathogens and then mark them for destruction. Some of the cells become memory B cells because remember, your B lymphocytes and your T lymphocytes have the ability to produce a memory. So here you see some of the cells actually become memory cells that are primed to respond to the same antigen if that infection should come back again. This picture really ties in a lot. It is busy, but it pulls together everything we've been talking about up to this point. Different ways that your body can respond to a foreign invader. And this is called the simplified summary of the primary immune response. Here's your antigen at the top, and that antigen comes in, and the first thing it runs into is your innate defenses. Skin, mucous membranes, and if it happens to get through that barrier, the skin, and then past the mucous membranes, then you have your phagocytic cells, you have your antimicrobial proteins, you have fever, you have inflammation, and then if it gets past that point, sometimes it's eradicated here, But if it goes to the point where your adaptive immune system becomes activated, you have this piece over here in pink, this over here in green. And the pink is your cellular immunity, okay? That's your T lymphocytes. Humoral is over here in the green. That's the B lymphocytes. So it can go either way. Free antigens, let's go to the right first. Free antigens may directly activate a B cell. Not necessarily the most common way, but it can happen. And so the B lymphocytes are activated. They form a clone. They produce plasma cells. Some become memory cells. They secrete the antibodies, and the antibodies are what's going to be involved with complement. Remember we talked about complement, to mount up a chemical attack on the antigen. So this is the B lymphocyte and how it does its job, mainly in the production of antibodies. Now, you could have that particular pathogen to be ingested by an antigen, by a cell, either an antigen infected body cell engulfed by a dendritic cell, or just to be ingested by an antigen presenting cell. And that antigen presenting cell presents those complexes, and this is MHC class 2, presents it to your CD4 cells, and those are your T helper. Or it can go this route, and it can go activate cytotoxic T cells. And if it binds to a cytotoxic T cell, then it can activate them to mount up a response against cells that are infected. Some of them become memory cells, and knock out any cells that are infected by, say, a virus, or if it works in response to cancer cells or parasitic infected cells, that type of thing. This route here, take a look at this, because if you activate your T helper cells, the T helper cells are at the hub, and they can activate non-specific killers, your natural killer cells, your phagocytic cells to engulf these foreign substances. They can also co-stimulate cytotoxic Ts. They can co-stimulate your B lymphocytes. So they are in the middle, and they're the ones that can actually draw in all arms of the defense. So those are the guys that are really important. You can get some activation independently of the cytotoxic Ts and B lymphocytes, but this is really how it works in most cases, is activating the T helper cells. So spend some time just looking at this and seeing how this all ties in together, because it really shows you how all arms of the defense are integrated in many ways. So now I want to look at antibodies a little bit. So antibodies, remember, are involved with the B lymphocytes. This is what they look like. They look like Ys. They have a heavy chain, that's the longer polypeptide chain. Then the light chains are the shorter ones. You have a constant region, okay, which is this part here, the stem region. And then the variable regions are up here at the tips. So all of your antibodies look like the classic Y, but the thing that's different is this little region right here. And it's like if you have a key, the teeth of all keys look alike, but the teeth of the key are unique to whatever that lock is. All these Ys look alike except for the tip, and that's where the teeth would be, where there is some variation. So that's where you see these antibodies differ, is in this variable region. These little yellow things here, these are disulfide bonds, and disulfide bonds help with attaining that three-dimensional structure characteristic of all antibodies in general. So there's 5 different classifications of antibodies, and we'll just go through these briefly. But IgD is the one that's actually attached to B lymphocytes, where it functions as the antigen receptor on B cells, okay? So IgD attached to B lymphocytes. IgM is a pentamer, meaning that it has 5 antibodies that are joined together in a central structure that looks like a pentagon. And this has a lot of binding sites, because each antibody has 2 binding sites. Well, because this has 5 antibodies, you have 10 binding sites. So it is something that can cause strong agglutination. This one actually fixes and activates complement, and it is the first one that you see in a primary immune response. It's the one you see most often. And because it can form large agglutinating complexes, it's used in a lot of your kits. Like if you were going to have a monoclonal antibody that's used, like say in blood typing. When you did the blood typing exercise in lab and you're looking for a strong agglutination reaction that you can see with the naked eye, these are the guys you want to use. Or quick strep test or mono test that are used in the lab makes a visual something that you can visibly see. So that's IgM. Now IgG is the most abundant and diverse antibody in the plasma. It accounts for 75 to 85% of all circulating antibodies. This is very important. It protects against any type of infection, so bacteria, viruses, toxins. It does fix complement, so you want to remember IgM and IgG can fix complement. And it's the main antibody of both the primary and secondary immune responses, and this one does cross the placenta and confers passive immunity from the mother to the fetus. So the most important one is IgG. Now you go to IgA. IgA is a dimer, and you see that it has 4 binding sites, and the 2 antibodies are joined together by their stems. This is the only one of the 5 classes that's found in humors of the body other than blood. So it's found in saliva, sweat, intestinal juice, milk, and it helps to prevent the attachment of pathogens to epithelial surfaces because it is released in sweat. And it does protect, like, it's found in the tears of the eyes and the eye surface. It helps to protect you there. So very important antibody is found in breast milk. So it helps to give some immunity to the baby. Babies that are breastfed get this naturally occurring antibody. So that one's very important too, but remember, found primarily in fluids other than blood. And then IgE is the last one, and it is a monomer, but it's a little larger than IgG. And this one is secreted by plasma cells in the skin, mucosa of the GI tract, respiratory tract, and it is one of those that can be involved with an allergic reaction. So also involved in parasitic infections. So you'll see this one involved in hypersensitivities, and we'll talk about this at the end of the chapter. So the defensive mechanisms of antibodies. Remember, antibodies are proteins. They don't destroy anything themselves. Okay, so they're not alive. They inactivate something and they tag it for destruction. And here's the plan of attack. Here's the 4 things that they do. And if you remember these 4 letters, PLAN, plan of attack. Neutralization means that it can block binding sites on viruses or exotoxins and inactivate them and keep them from gaining entrance into cells. So that's neutralization. Complement fixation, remember that MAC attack complex, the membrane attack complex, causes lysis, so there's the L. Agglutination, cross-linking of antibody-antigen complexes, causes these larger clumps to form that can be ingested by your macrophages. And precipitation, substances that would be soluble, cross-linking them into insoluble complexes, and this is all because the antibodies have 2 binding sites, so you can cross-link different antigens together and make them into larger complexes. Now, the primary immune response is the very first response to an infection. So during this time, cell proliferation and differentiation happens upon first antigen exposure. This takes time for your cells to become activated and mount up an immune response. So the lag period is around 3 to 6 days. Peak levels of plasma antibody are reached in about 10 days, and then your antibody levels decline. So the first time you have infection, you get the malaise, you don't feel right, you're not sure what's going on, you start to feel the fatigue, the tiredness, then you get the aches and whatever else are the manifestations of that illness, but it takes a while for your immune system to respond. Now, the secondary response is if you have a re-exposure to the same antigen. This response is faster, more prolonged, and more effective than the primary response because you have memory. So the sensitized memory cells respond within hours, and the antibody levels peak in 2 to 3 days at much higher levels, and antibodies bind with greater affinity. And the antibody level can remain high for weeks to months. So this secondary response, if everything's working fine, it'll mount up a response so quickly that you will not get the illness. So that's why the memory is very important. Here it is in picture form. So it's showing you the primary immune response to an antigen. You have that lag period of 3 to 6 days. Then you develop the antibody titer. This graph here is showing you the relative amount of antibodies, just a relative number. So within the first week, and then after 2 weeks, 3 weeks, you start to see the subsiding of the antibody, but you still have your memory cells. And if you have a secondary immune response to that same infection that you encountered here, then look at the titer. It looks almost like a J curve going up. And within probably 24 to 48 hours, you have a much larger amount of antibody produced, and chances are you're not going to get the infection. Now, if you have an exposure to something totally genetically different, you have to go through the whole process again of the initial primary response, development of the antibodies, and production of the memory cells. Organ transplants. Now, there are 4 varieties of types of tissues that you can transplant from one person to another. Sometimes, if at all possible, you would want to take tissue from the same person and transplant it somewhere else on that same person, like, say, a blood vessel. If somebody has a blockage in a blood vessel, to take a graft from another part of the body, from a limb, and use that person's own tissues, because then you're minimizing the chance of any type of rejection. Sometimes people, if they know they have a surgery coming up and they may need a transfusion, they may do autologous donation, have a unit kept on in the lab for use in case they need it. So that's an autograft, okay, from one person to the same person, but from a different part of the body in many cases, or like skin. If a person has a burn, they can take skin from one place in their body and use that somewhere else. Now, an isograft, if you have the luxury of having an identical twin, you want to remain friends with that person because you never know when you're going to need some parts. I always say that as a joke, but isografts are between identical twins because they are genetically the same. So if you have that possibility, then you can get tissue from one person to another, but that's not not so common. Allografts are the grafts between individuals within the same species who are not identical twins. These are the hardest to work with because you have to have a reasonable amount of genetic continuity between those major histocompatibility complexes in order to transfuse tissue or transplant tissue successfully. And it's around 70 to 75%. It has to be this genetic similarity is very, very close, or else you're not going to get a successful transplant. So allografts are between people that aren't genetically the same, but the most difficult to deal with. Now, xenografts are from another animal species, and they are working with, you have bovine transplants, porcine, pork, cow, different types of tissues they're working with that they have successfully used and integrated enough genetic markers to make them so that they aren't rejected like you normally would think they would be, but they're using that quite a bit, and they're continuing to develop even more and more similarity between different species of animals. So make sure you know these 4 terms and how they're defined. So when you do have if you do have a transplant, you have to, unless it's an identical twin, you're going to have a problem or a potential problem for rejection for the rest of your life. So you have to be treated with immunosuppressive therapy. to suppress the immune response, the inflammatory response, because remember earlier we said that inflammation is the body's SOS signal to alert the body and immune system that there's something wrong. So you want to suppress inflammation. So corticosteroid drugs are one of the most common medications or regimen of treatments that's used to suppress inflammation. So that's one of the things they use quite often. And anti-proliferative drugs to keep the immune cells from proliferating and forming a clone against that transplanted tissue. And then immunosuppressive drugs. And the cells that are the culprit are your cytotoxic T cells. Those are the ones that recognize MHC class I markers that all your body cells have that are foreign. So those are the cells that cause you the problem. Many of these treatments that are immunosuppressive and anti-proliferative are very toxic substances, and they do have severe side effects on their own. But if you need a kidney or a liver or a heart to survive, you take your chances, you take the transplanted tissue, and if it buys you 5, 10, 15 years, it's well worth it. And they're improving on a lot of these treatments too. Now this is looking at hypersensitivities. So we'll briefly go through these, just generally speaking. But there are 4 types of hypersensitivities, and hypersensitivity is another name for these, just simply allergies, and not everybody has them, just some people have them. And the one that you see quite often is a type 1 or an acute hypersensitivity. And this one begins almost immediately after contact with the allergen. And what the problem is, remember we talked about different classes of antibodies when we mentioned IgE? Well, IgE is the antibody that's involved here. So the first thing that happens is you're exposed to the allergen. And then you have cells that are going to be stimulated, and they're going to produce IgE. IgE is going to bind to basophils and mast cells. Now, if you remember, basophils and mast cells have granules that release histamine. So the first thing that happens is this sensitization stage. That's setting the stage for the next exposure. So the first exposure, no problem. Now, the next exposure, the stage is already set. And the antigen now is going to bind to these cells, these mast cells and basophils attached to the IgE. And those cells, once they are activated, start to degranulate, and they start to release a lot of histamine. Histamine causes blood vessels to dilate and become leaky, which promotes edema, stimulates secretion of large amounts of mucus, and causes smooth muscles to contract. If respiratory system is the site of the antigen entry, asthma may occur. So mild cases, you might wind up with some nasal congestion, or mucus production, when you have like an allergic reaction or sinusitis, that type of thing. And there's a lot of people that have this, and sometimes it's just seasonal. The problem is if you have a severe reaction, this is called anaphylactic shock. And it's in response to an allergen that directly enters the blood. And the basophils and the mast cells are enlisted throughout the entire body. And systemic histamine releases in massive amounts may result in constriction of the bronchial passageways as a whole, sudden vasodilation, and loss of massive amounts of fluid from the bloodstream that results in hypovolemic shock and death. And the treatment for this is epinephrine, which reverses the vasoconstriction, opens up the airway, and causes constriction of those blood vessels. So some people that have allergic reactions or allergic problems, like say penicillin, shellfish, things like that, peanut butter, they may carry EpiPen with them just in case they do have an exposure. Now the type 2 reactions are called cytotoxic. This is where antibodies are developed that bind to antigens on specific body cells. So you develop antibodies that start attaching to your own tissues, and stimulate phagocytosis and complement-mediated lysis of cellular antigens. So an example of this would be mismatched blood transfusion reactions or hemolytic disease of the newborn, what happens with an RH mismatch. Type 3 is an immune complex hypersensitivity. So the antigens are widely distributed in the body or the blood, and so antibodies are formed that make insoluble antigen-antibody complexes. You get agglutination, and the complexes cannot be cleared from a particular area of the body. And so the immune system responds against your own tissues, and it causes intense inflammation, local cell lysis, cell killing by neutrophils. And if you see the description here, you're talking about autoimmune diseases. So type 3 is autoimmune. And you think about things like lupus, rheumatoid arthritis, those types of things fall into that category. Now, delayed hypersensitivity, or type 4, is slow onset, 1 to 3 days. Normally is something that's topical and works through the skin. And this mechanism depends on T helper cells. Cytokine-activated macrophages and cytotoxic T cells are the ones that really cause the problem. And things like allergic contact dermatitis, poison ivy, the agents act as haptens. They don't develop this reactivity. It's more just like a reactive type of reaction that doesn't involve stimulation of the immune cells in a normal pathway. We talked about haptens in the first half of this chapter lecture. So what you do is, if a person has this type of delayed hypersensitivity, is you kind of do a skin test. And you see what kind of substances the person reacts to. And sometimes it could be 60 to 100 different agents that they're looking at, and they'll do little pricks of the skin to see what type of substance the person might have a problem with. The substance the person has a problem with. So autoimmune diseases. This is where the immune system loses the ability to distinguish self from foreign substances. And you get the production of autoantibodies, and your sensitized cytotoxic T cells destroy body tissues. So some of the examples: MS, myasthenia gravis, Graves' disease, type 1 diabetes mellitus, lupus, glomerulonephritis involving the kidneys, and rheumatoid arthritis are some examples, and there's many, many more. The mechanisms of autoimmune diseases: you have ineffective lymphocyte programming. You get self-reactive T and B cells that should have been eliminated in the thymus and bone marrow that escape into this circulation. This is one theory, one way it could happen, that when they were being programmed, the ones that were anti-self and should have been destroyed somehow get out of jail before they're destroyed, and they go rogue in the body and cause problems later on. Another thing that can happen is new self-antigens appear. The antigens change because gene mutations cause new proteins to appear, and your immune cells are programmed to look for a certain sequence of protein, a certain MHC complex. So if it's genetically altered, now it's not self anymore. So changes in self-antigens can happen by hapten attachment, these allergic proteins, or as a result of infectious damage over time. The third thing that can happen, if the determinants on a foreign antigen resemble self-antigens, then antibodies made against the foreign antigens cross-react with self-antigens. So these are different ways that your immune system can be fooled into attacking its own tissues, or your immune cells that are anti-self go rogue and start to cause you problems. So all kinds of things that can happen, unfortunately. And not everybody reacts, remember. We're all a little bit different. And these are just some theories of how autoimmune diseases actually are started. So congenital immunodeficiencies. Some people are born with the inability of producing a certain type of immune cell, whether it be a B cell, T cell, and sometimes bone marrow transplants help. But severe combined immunodeficiency, or SCID, SCID syndrome, is a genetic defect where you do not have the ability to make B or T cells. There's a problem with an adenosine deaminase. It's an enzyme that enables metabolites to develop that are lethal to T lymphocytes. And this is what happens in this case, but the cells don't develop. And it's fatal if untreated, but like I said, bone marrow transplants have been very helpful. Acquired immune deficiency syndrome, or AIDS. This particular disease is important, and I'm only going to mention it briefly here because what it does is it interferes with the activity of T helper cells. This virus invades the T helper cells. And if you take out the general, remember, you've taken out the immune response. So what happens is that people that have AIDS, they actually don't die of the disease. They die because their immune system is suppressed, and they become vulnerable to opportunistic infections that most people don't get. Things like a fungal pneumonia called pneumocystis pneumonia, and also a fungal infection called Kaposi sarcoma that causes disfiguring ulcerations of the skin. A lot of times they occur on the face. So the problem is the eradication of the CD4 or T helper cells and leaving yourself wide open to other infections. So that ends chapter 21.